Good afternoon, guys. Today our topics: object genetics, mode of gene expressions, and various and depends on properties of genes. What is genetics? Genetics it is science about heredity and diversity. The heredity and diversity and fundamental properties of living things. The science studying laws of heredity and diversity refers to as genetics. The genetic studies of the following problems. Next problems. It is storage of genetic information, transfer of genetic information, realization of the genetic information, and change of the genetic information. Chromosomes are subdivided on autosomes and heterochromosomes. It is means sex chromosomes. In 1924, under the author of Levitansky, the diploid set of chromosomes of a somatic cell has been named karyotype. It's characterized by number, shape, and size of chromosomes. Under Novatian's author, chromosomes are placed accordingly their size that have name ideogram, systematized karyotype. In 1960, the International Classification of Chromosomes there suggested chromosomes classification based on size and arrangement of centromere has been offered in Denver. In karyotype, a somatic cell of human distinguished 22 pairs of autosomes and pair of sex chromosomes. Guys, on these slides you can see human karyotype structure. Please, female structure karyotypes. Please, you should transfer it is diagrams in your sketchbook. It is your homework. A set of chromosomes in somatic cells have name diploids, and in sex cells haploids. It is equal to half of autosome set. The human karyotype was divided into 7 groups, depending on the size and shape. A groups. First 3. Biggest metacentric. B. 4/5 groups. Biggest submetacentric. C. 6/12 groups. X chromosomes average metacentric. D. 13/15 groups. Average acrocentric. E. 16/18 small submetacentric. F. 19/20 small metacentric. G. 21/22 pair, 21st, 22nd pair, Y chromosome, small acrocentric. Now we're talking about general science, genetics subjects. It is heredity, diversity, inheritance. What is heredity? Is a property of living things to transfer morphology function and individual development traits from parents to offspring. Diversity is an ability of progeny to be distinct from parents by morphological and physiological traits and features of individual development. Inheritance is a way of transfer of a genetic information through sex cells at sexual reproduction or through somatic cells and asexual reproduction, a material basis in ovary cells and spermatozoa or somatic cell. Please, next science living things. It is heritability. Is a degree of ratio between heredity and diversity. Gene is unit of heredity and diversity. It is the sequence of DNA molecule giving the information of synthesis of certain polypeptide, the set of alleles and an organisms which receives from the parents refers to as the genotype, the set of genes in haploid sets of chromosomes termed a genome. Set of all external and internal traits of an organism refers to phenotypes, and a separate traits refers to phen. For example, the nose form, the auricle form, foreign fingers or foots, hands, color of hairs are external phenotypic traits. Features of a structure of a stomach, the contents of leukocytes and erythrocytes and blood are the internal phenotypic traits. Mode of gene expressions in various and depends on properties of genes. The gene in discrete in the action provide curves of a certain biochemical reaction, a degree of a character expression of repression. A gene is specific in the action in the responsible for synthesis of a primary structure of a protein's molecule. The gene can have a multiple action. It is named pleiotropy. The different genes which are located in different pairs of homologous chromosomes can act on development of the same character, intensifying or weakening in polymeria. What is genetic material? It's components of a cell which have structural, functional unity and provided storage, realization and transfer of the hereditary information, and a sexual and sexual reproduction. The genetic material possesses universal properties of living things. Discreteness, continuity, linearity, and relative stability. The basis property of genetic material, the genes, is storage and transfer the hereditary information. The gene is capable to change the genetic information due to mutation. The gene is capable to repression and the transfer on next generation. The gene is capable to realization protein synthesis. The genetic material has stability. Stability of genetic material is provided by diploid xenochromosomes, double helix structure of DNA, redundancy of genetics code, repetition of some genetics, repairing of DNA broken structure. The site of a gene carrying the genetic information is termed exon, and the site of a gene do not carrying the genetic information is termed intron. For example, the globulin coding gene has 3 exons and 2 introns. The main position on this theory is that there are 2 types of genes in DNA molecule: structural and regulatory. Structural genes encode proteins that are used in metabolism of biosynthesis or that play a structural role in the cell. Regulatory genes are genes those product either RNA or proteins interact with other sequence structural genes and affect their transcription of translations. In many cases, the product of regulatory genes are DNA binding proteins. Please, on these slides you can see stages of a differentiation of a sex in human. Please, for example, the periods intrauterine development, weak, and development of gonads and sex characteristics. Please, 3 week primary sex cell, 5-week formation of primary gonads, 6-week basexual gonads, 7-8-week development male gonads, testicle, 8-9-week development female gonads, ovary, 7-9-week formation of a hormonal sex, 10-12-week formation internal genitals, 12-20-week formation external genitals. Please you should it this timetable, transfer in yourself sketchbook. Rudiments of gonads and early embryos style 5th of 6th week do not differ at different sexes and will consist of an external layers, cortex, and an internal layers, medulla. Primary cells of a germ tract are found out in human on 3rd week of embryo developments and ectoderm of Volk sac. Then, under influence of hematocytes, signals will migrate in gonads. This migration does not depend on sex. Rudiments on gonads can develop in ovary or testicle. The differentiation of male gonads is observed on 7th week to 36th day of development, when testicle starts to release androgens which determine the male development. Development of female gonads is observed on 8th week. Result of that is formation of female hormones, estrogen. Please, our lecture's finished. If you have questions, give me questions. I am ready all times answer these questions. Please reading, prepare these lectures on next classes. And who is one, please will give me